So lentivirus vector manufacturing using suspension cell line. All right, so here is the content for today, the viral vector in gene and cell therapy. The starting materials like plasmids used for lentiviral vector packaging, and the lentiviral vector with the adherent manufacturing, and then the suspension manufacturing, and last but not least, would like to talk about GenScript ProBio track record. Here, over the last few years, gene therapy has emerged as a promising medical tool to treat different kinds of diseases. This novel approach is underpinned by the positive clinical results achieved in patients, followed by regulatory authorities' approval in vivo products commercialized in both US, Europe, and in China. Recently, a few companies, for example, like Bluebird, Kite Pharma, GlaxoSmith, Unicor, has been approved by the US FDA relatively in 2017 and so on, and also by the European Medical Agency. These milestones have been considered as breakthrough in the medical field and have encouraged pharmaceutical companies to actually increase investments in gene therapy products and developments. In 2018, FDA have actually received 206 gene therapy related IND submissions. Here, I'd like to talk about the market perspective. Lentivirus have gained value over recent years as generic carriers in gene therapy. These viral vectors are safer than what was previously being used for gene therapy. These characteristics of the lentivirus is ideal for clinical research and has been demonstrated with the approval of lentivirus-based gene therapy. And this characterization of the LVV have gained a significant improvement from the recent years. A large actually number of functional lentivirus particles are required for clinical trials. As we can see here in the chart, you can see in the chart about the 2018 and in 2021. The gene therapy related was 49% and cell therapy was over 40%. So overall, 40% of the cell therapy programs in clinical phases, large amount of LVV is required in the future for clinical phases, as they are targeting solid tumors and other kinds of cancers. So this is a very exciting news. And even in the clinical phase, you can see 43%, and here is the 57%. And whereas in the R&D phases, it's 56%, and there are 45% for the blood tumor diseases. All right, here are, I would like to talk about a few challenges in the process development of a viral vector. And here, GenScript have actually a one-stop solution for plasmids to lentivirus vector to solve our industrial bottlenecks. Here you can see that there are a few things I would like to mention about is the temperature sensitivity, the acid base, the salt sensitiveness, the low tolerance of shear force, which is actually the biggest bottleneck, the most important factor to actually consider. And then we have also a few issues like large-scale transfection using different transfection reagents and to control the titer ratio and the loss of strial filtration due to the different sizes. Therefore, the first of being focused on solving these mentioned drawbacks, which is associated with the production and purification of lentivirus under good manufacturing practices. Actually, in recent years, we have seen the development and optimization of new protocols, packaging cell lines, different culture mediums, which is very important to actually optimize. Here now I would like to mention about our plasmids for lentiviral vector manufacturing. The good news about it is like we have been registered with the FDA, CBER, we have the number, the reference as well, which will be immediately available to support cell and gene therapy products. The readiness for the BLA will be 2021. And here we have a screenshot we have mentioned over here for the acknowledgement. And would also like to talk about the advantages of our Lenti helper plasmids. We use the third generation, which is idle for many different things. For example, like reducing the time and cost by using the 3 helper plasmids, which is simplified regulatory documents as well, so it's easier to access, and completely and immediately available for preclinical and clinical manufacturing. And it comes with a very clear history. And the most important thing is all the 3 plasmids comes without any IP issues. So that's also one of the most important feature of our plasmids. So now I would like to talk about a little bit about the Adherent manufacturing. Give you a short introduction about it. Here is the map, the flow chart, how it's done, the cell thawing, the cell expansion, the cell factory, then transfection, then harvest, clarification using different filters, then TFF, then the nucleus treatment, then we'd go for a different chromatography, then sterile filtration and TFDF, and then fill and finish. And here, the most important thing, the characteristics of the QC testing, which you can see here on the right side. It's this assurance that each batch meets. You have to be more specific with the purity, the potency, the safety, and identity. Therefore, the QC testing should be precise and accurate to provide the clinical investigation team and the regulatory team to understand how it works. One of the challenges of the QC testing is the viral vector is a high degree of complexity, as it's a class of biologics, which I have marked here in the yellow, which are very important CQAs. We use different kinds of tools, which are very crucial in process development, and it plays a very, very important role in the downstream process. And furthermore, the development of the faster, more accurate tools, like the assays, which you can see in the thing, is needed, and it's time consuming. And especially we usually go for qPCR, which is usually the method of choice. Droplet digital ddPCR has been recently used for quantifying viral genome. And ELISA is also widely used in techniques for viral antigens. And here you can see I've highlighted 4 more important things. The BCA, which you're going for the adherent part, the BCAs needs to be removed when it comes to adherent service. And the method we use is the ELISA. And the impurities, the HCP or HCD, it's by the SCP will be for the ELISA, and with DNA with the RT-PCR. And the functional titer with FACS, that's where we do our testing as. Now coming to the suspension part, a lot of people do have questions about what are the basic differences between suspension system and adherent system. We do get a lot of questions, and we always try to answer, but here I have classified into two different groups. Especially when it comes to the suspension system. Here we use inline system, and we do also use the commercial cell lines as well, which comes with a very high license fees. So we kind of use our in-house built system, especially with HEC 293 or 293T. At the other hand, when at Adherent, we use 293T cell. And with the suspension system, we use bioreactor, which is serum-free. And in Adherent system, you have to use the BCA. The serum is there, and it requires to be removed. And with the suspension cell line, you get a very high titer in the supernatant, which is a very positive sign. And in the adherent system, you get lower titer in the supernatant. And the high batch yield in the suspension and lower in the adherent. And no residual BSA. So it's a suspension free, so of course there's no BCA impurities, so you don't have to do an extra removal step to take it out. But it comes with the higher HCP content in the supernatant. But in the adherent, there will be very low HCP content in the supernatant. When it comes to purification, there are a few different steps, like especially for clarification, you use different techniques, but it's somewhat similar to the suspension system. So TFDF and then chromatography. And I also would like to say a few more things about it. Is like the adherent system is laborious. It requires a lot of space. It takes time. So it's much, much feasible to go for the suspension system. So as I've mentioned that we have our in-house cell line. So it's Power S, which is trademarked, is 293 cell. So here in the chart on the left-hand side, you can see the crude titer FACS TU per ml, that the Power S293T, which is GenScript in-house cell line, have a higher titer compared to the commercial cell lines. And on the right-hand side, we have did a few experiments with gene of interest, GOE 1, 2, 3, 4, 5, and 6. And you can see that it's almost the same with a higher crude titer, which is 10 to the power 7, 10 to the power 8. So to conclude, I'll say it's better or similar performance of our Power Cell 293 cell line compared to the top vendors, which is available in the market. We do get higher titers, and it's consistent and stable yield when we compare it with different GOI. This is the very important part when it comes to consistent and stability. Here, I would like to talk about the scalability and the process. We have used different projects, and we compare it different using facts for our thing. So in project 1, you can see when we have used 2 liters to 10 liters. We have used numbers of batches, and here you can see it's consistent, and the crude titer, it's the results are really, really great. And you can see the comparison between the project 1 and project 2. So it's almost stable. And with on the right-hand side, you can see number of batches of 3 for project 3 and 4 for project 4. So it's almost consistent. Okay, the third highlight is about the high T transduction efficiency, which we have compared it with few other vendors in the market, vendor A, B, and with GenScript Pro Bio, which is Power S293 cell. So it's almost 20% plus, so that's high performance in T cell transduction, comparable with Adherent process. Here with customer B, we have GOI1 and GOI2, and the CAR-T percentage is over 60%. And in the GOI2, it's over 40%. And the highlight 4 is the superior yield and the cost. Here with the titer improvement, we have received 40-fold increase when it comes to suspension culture. So here on the right-hand side of the chart, you can see the adherent and the suspension. The chart, you can see that it decreases 50 to 60%. The cost reduction is 60% when we operate on suspension culture compared to adherent. And here you can see the yield increases too. Like with the 10-liter batch, it increased 4 times. When we have run the batch in 50 liters, it was increased 5 times. So undoubtedly, when suspension process is much, much better compared to adherent when it comes to higher yield and the price reduction. So drastically drop in 50 to 60%. Okay, so I would like to talk about the key points in the process development of lentivirus manufacturing with the suspension culture. Here, when you actually consider about the process development, you have to consider a few things. For example, we have to see which cell lines are you going to use, whether you want to use it for the adherent culture or you want to go with suspension culture. What will be the ratio for the transfection? You want to use a stable cell line, or which cell line you wanted to use. And when it comes to requirements specifically for the raw material and other products or regulatory and quality when it comes to different materials. Which is very, very important. You actually need to know what kind of material we can use to get the optimized result. When it comes to filters, when it comes to resins, when it comes to the fiber for UFDF or TFF. So these are very important points you need to consider. The crucial parameters, I would say, and then for the scaling up and how it's going to be transferred, how will be the titer, and what will be the cell density and how are you going to control your turbidity, and how are you going to reduce your impurities, and what plan do you have when it comes to sterile filtration. You're going to lose a lot of virus in that process. So here, the upstream process for our cell line. Here I have a few things like the cell line, the packaging system, the serum film medium with VCD and VIA. I'll start with the cell harvest. Actually is the interface between upstream and downstream. The time of harvest should be determined to take into consideration different parameters like quality, the product quality, not only productivity, the product stability. For example, like what's the cell density and what's the cell viability, and also the enormous impact for the later on steps like purification steps. The higher increase in host cell protein, the host cell DNA, and the cell debris. For example, that will be a burden for the downstream process for the purification. However, there is actually no idle time for time of harvest, and you need to adjust the parameters, and you have to take that in account when you are doing the development as manufacturing. And for example, here to wind up, it's like 4 things: the cell density at the transfection, which transfection reagent is going to be used, what will be the plasmid ratio, the ratio between transfection reagent and total DNA. So you have to optimize all these processes, all these parameters to get an ideal answer. So this is the case study which we are talking. So here is the 2 graphs. As in the graphs, you can see the stability of our process in which different projects were involved, and the facts has followed the similar higher recovery pattern. Here you can see in the projects, 5 different projects. On the other hand, on the right-hand side, is number of batches which we have exceeded, no matter if it's a small or large scale. We have received the same results, and when it comes to scalability, it was stable as it looks. For example, we started with 30 ml to 1 liter to 2 liter to 10 liter, and these are the number of batches to optimize our process. And here at the bottom, we have compared it with 2 liters versus 10 liters. And here you can see the trend is almost similar when it comes to 2 liters and 10 liters, which will only get you when you are doing many experiments and you're optimizing. So let's see, for example, when it comes to 30 ml, 26 experiments were done. When it comes to 1 liter, 30 experiments were done. And then only we understand like this is very stable and we are getting good results. All right, here on the 50 liters, a case of a 50-liter batch in a single-use bioreactor. And here with, again, 2 graphs, and you can see the crude titer. When it's 50 liter, 2 liter, 30 ml, 2 liter bioreactor, and 30 ml shaker flask, which we have measured with P24 and the crude titer as well. I would like to brag about our 50 liters actually. The data says it all, and we have achieved good results and proud to share it with everyone. We have actually successfully reached a higher crude titer in most of our batches. Okay, here is a very, very important slide. I would like you to please pay attention here as well. The downstream process, which is very, very challenging and a lot of things to consider. So all of these slides, which you can see the data for the downstream process, just want to tell you guys that if you want to do each process study, what the CPPs are, the CQAs are, we should take the consideration first. For example, the nucleus treatment, the selection of filter, chromatography, the TFDF, and the sterile filtration as well. And actually, these kind of things will become the pillars of downstream processes. Ultrafiltration is the method of choice for concentration and difiltration. For these particles will remove low molecular weight impurities and reduce the handling volume. For example, that if you are talking about TFDF, the molecular weight cutoff is between 100 to 500 kDa. The membrane cassette, which is from 100 to 300 kDa. For example, the use of hollow fibers devices can result in lower shear rates when compared to the member cassettes devices, which is usually used in manufacturing of several viral particles. When it comes to different chromatographies, we have gained more importance in the manufacturing. For example, like IEC, ion exchange chromatography, is one of the most widely used techniques for purification of large particles, which exploits indifference and it's particularly different charges as well. And then the ion exchange chromatography technique can be applied depending on the net charge of the particle. For example, if it's LVV or AAV or adenovirus and so on, a difference. The net charge, the pH will depend on the particular surface. Most of the viral particles have an isoelectric point below 7.4, negatively charged. Lentivirus is also an example of a viral vector that has been successfully purified using ion exchange chromatography. We have seen it in different companies and our company as well. The membrane filter is a key parameter in sterile filtration. When it comes to sterile filtration here, it's actually the losses are mostly in the sterile filtration. So here, the basic things which I have mentioned here is the temperature. When you are talking about nucleus treatment, the temperature, the pH, incubation time, final concentration of benzonase, the HCP removal rate, plasmid DNA removal rate, recovery rate. And when it comes to the filter, the pressure, you have to check what's the pressure when you are selecting a filter and what's the turbidity. And the most important thing is the area you're going to use, because when you are using the small scale, you are using the small scale or you're using the large scale. And the recovery rate, the SCP removal as well when you are using different filters. So here we have seen the trend when using different filters, the recovery rate on different steps. And now I want to talk about the chromatography. When it comes to chromatography, the parameters which is important is the loading volume, like how much loading volume, how much buffer you're going to load, what will be the flow rate, and what is the recovery rate and the removal of the impurities. And the TFDF, the most important thing about it's the pore size. You have to know what kDa you're going to use for different viral vectors and what shear force you're going to use. Recovery rate and the removal of HCP and SCD. And then about the sterile filtration is the same as when it comes to selecting filters. You have to see the pressure, you have to set the flow rate, and you have to check for the recovery rate, because maybe you will be losing a lot of vectors, a virus in the filter. So it's a very low recovery. There are a lot of batches we have done, and the recovery, it's here the percentage we have when we have loaded the supernatant, and with the step 1, 2, step 4, and step 5. So it's a good recovery when it comes to FACS, and it was measured by P24. And here, the HCP removal, the highest when we have loaded the supernatant, and when it comes to the step 5, it's within our target. And with the HCD as well, it's super high. And when it comes to our last step in our process, it comes to very low. Okay, lastly, I would like to talk about our GenScript track record. Why should you choose us, and how you can put the trust in our company. We have been collaborating with a lot of different reputed companies around the world. And our mission is very clear, that we are very dedicated to providing end-to-end services, very professional, and help our customers in any sort of ways. Here, I would like to also show our solid track record, that we have 6 IND approvals from the local body and the FDA, over 20 P&V CMC projects running, and 30 clinical plasmid manufacturing batches as well. And over 10 clinical lentivirus vector batches are going on. And we have been collaborating with some of the best companies in China and around the world. For example, I want to talk about XLife's TCRT, which have submitted the IND. They got the approval in 2019, and they have started their clinical trials. The same thing with STEMI-RNA. They got the approval in 2020, and they have started their clinical trials in April last year. And the final one, Abogen, the first batch was set up by the Chinese government in 2020, and they also have started in 06/2020 for the clinical trials. And the other parts I would like to mention is, like, we have global clients all around. And in China, US, Canada, Korea, and Japan, we have different ways to talk to our clients, and we have received 100% satisfactory outcome. For example, 25 CMC client audits in China, and 16 client audits from US, Canada, and Europe and Korea, 13 satisfactory, and 3 are ongoing. And the IND filing track record is that with China, NMPA, which is the local body, and FDA for plasmid and virus, we got approvals as well. And within US too, because our head office is in US. And 20 ongoing plasmid and virus CMC projects. So the reason why we are sharing this is, like, if you guys are choosing us, it's more like you're in the good hand, and we have a professional, dedicated team to cater your needs. For example, if you guys have any questions regarding our suspension process or anything, would be glad to help you and assist you. And that's all for today's webinar. And if you guys have any questions, we would be glad to take. Yeah, Casey.
Okay, thank you, Dr. Ali, for the wonderful presentation. So we'll move on to the Q&A session right now. The first question, if we use suspension manufacturing, what's the license fee for using the Power S293T cell line?
Okay, when it comes to the Power S, yeah, it's a good question, because that's our in-house Power 293 cell. You are supposed to pay a license fees based on the payment milestones, For example, we have set like firstly at the IND approval, secondly at the clinical phase 1, and lastly when it's a BLA. With our license fees, it's quite low compared to with our other competitors in the market. Yeah.
Okay, thank you, Dr. Ali. So the next question, what's the advantages of PowerS293T compared to other companies?
The Power S, as we have seen in the slide, that it gets a higher yield, or at least similar to the commercially available cell lines. That's the first thing, you get a higher yield. And the concern about the license is that it covers IND, clinical, and commercial. And we have actually performed a lot of batches on our Power S, which has given stable, a stable FACS results. And we have used it with different GOI, and we have achieved a very high titer. So this is a best answer I can give. And then lastly, I would also say that the license fee for our Power S293 cell is relatively low, and you don't have to pay any royalty, or you don't have to pay any maintenance. So it's a lot of advantages when it comes to anyone thinking about getting Power 293T. Yeah.
Okay, thank you, Dr. Ali. So the next question, can we select cell line for manufacturing our LVV with suspension culture?
Yes, you can definitely select. There are a lot of different cell lines available in the market, and we do have our own as well. But if you guys wish to have your own, we can establish the MCB, WCB for you. We can send the detailed requirement to you. Please leave your contact, your information, how we can reach your email, so we can definitely contact whoever is interested. But we do have our own cell line, as you guys know, or we can use different commercially available cell line as well.
Thank you, Dr. Ali. So the next question, what's the main difference between adherent and suspension process?
Oh, yeah. The adherent and the suspension, yes. As I mentioned, like we do get a lot of these things. What's the difference between two of these systems? So adherent system, it's you have to use the serum, the bovine serum that is there. So in suspension, there is no BSA when you are using LVV production for the suspension. And the passaging for the suspension process is much more easier when it compares to adherent. And again, for the larger scale, as I've mentioned, for the larger scale manufacturing, suspension cell line is highly recommended. Because with the adherent, it's very difficult to go on a higher scale. You need to have a lot of wells, you need to have a lot of space, and it's laborious. So it's always, suspension is always the best choice. And again, with suspension, you get higher yield, and in adherent, you get lower yield in the supernatant. And the satisfying T cell transduction rate is much higher in suspension when it compares to adherent. And I'm sure the suspension cell line can always be your first choice if you are trying to go for lentivirus production.
Okay, thank you, Dr. Ali. The next question, can you illustrate a bit more about your general purification process?
General purification process, so I understand basically the downstream part. So what basically is there is like you start with the UFDF, and you use hollow fiber, or you can use the cassette. But for lentivirus, it's better if we go for hollow fibers, because you can actually control the shear force, and it's much more idle for lentivirus. But if it's an AAV, you can go for membrane cassette. And then you have to also understand like what kind of chromatography you guys will be using, what kind of resin you are using, how you design your experiment. You want to go for size exclusion chromatography, or you want to go for IEX or AEX. So this is very important. Most of the people are using IEX now, and we use IEX and size exclusion chromatography when it comes to our process. And then the filtration, the clarification, it's a little difficult, actually. It just looks like you're filtering, but you need to know what kind of filter you're going to use, what kind of material you're going to use, and how much is the recovery. You need to understand a little bit about the filters as well. The most important, I would say it here, is like you even have to understand what charge does the filter carry. Yeah.
Okay, thanks, Dr. Ali. So the next question, what is the percentage difference of transduction efficiency in CAR T cells between crude before and after at the end of the downstream process treatment?
I mean, when it comes to the things, I mean, again, it's the same thing. Like, see, people have to, see the difference between the adherent system and the suspension system. So when it comes to the adherent system, the transductancy is much lower. So it's always recommended that you use suspension to get a higher ratio.
Okay, thank you, Dr. Ali. So the next question, do you have in-house data to compare several suspension 293T in the market in terms of virus title?
Yes, we definitely do have, as in the slide I have mentioned, that we have done almost over 30 experiments on a small scale to large scale. So it's been constant. It's been constant, and it's stable. So it can definitely be shared. Please get in touch with us, and we can definitely be glad to share the data with it, which when we compare to facts. Yeah.
Thanks, Dr. Ali. The next question, what is the most difficult process to get high yield of purification? Compared with AAD DSP, why does lentiviral purification process shows the low recovery?
When it comes to adherent, right? Like, I mean, the question is why there's low recovery in adherent, isn't it?
Low recovery for lentivirus purification doesn't really...
In suspension or in adherent?
Doesn't really state.
Okay, I think this question needs to ask like it should be suspension or adherent, but I mean, when it comes to low recovery, I think most of the recoveries are lost during the purification process, which again, it have to be optimized. We have to use different experiments to design and to see where and which recovery and which filters we are going to use, and what are the recoveries for that. For example, you get higher recoveries when you're using cellulose filter. When it comes to PPE, then you might get a lower recovery. So you have to optimize depending on your project.
Okay, thank you, Dr. Ali. Maybe we'll take the last question before we wrap up the whole session. Yeah, so the last question will be, you mentioned that suspension process has a higher whole cell protein impurities compared to with adherent process. Is the downstream purification process is more tedious?
Yeah, very good question. Yes, so when it comes to suspension, this is a biggest challenge or most of the industries are facing, the removal of impurities, the HCP and HCD. So of course, it makes a lot of pressure for the downstream team to purify and to control the turbidity. So I think if you choose the right material, the right resin when you are dealing with chromatography, I'm sure this can actually solve a lot of issues when it comes to when it comes to impurities. Again, it needs to be optimized. It needs to be checked where and how it can be helped, and especially the nucleus treatment will help you to optimize as well. Like some of the projects we have done it like after the harvest, in some of the projects you do it in the middle of the process. So you just have to optimize and see. It's a still very challenging problem when it comes to the impurities, which is HCP and HCD.
Okay, thank you, Dr. Ali. So due to time constraint, we'll just end the Q&A session here. We still have a lot of questions that we didn't get to answer, so I will email back all the answers from Dr. Ali to you. So thank you once again, Dr. Ali, for the great presentation, and also to all of you who joined us today. I hope you find this session useful and beneficial for your work. Okay, so thanks everyone for joining us again, and have a nice day.
All right,
thanks
thank you everyone.
Bye-bye. Bye.